and then still talk about it. So we'll try to make that happen. I wore my tweed today, so we can maybe make the biology happen. The start of the background information on our little science class here is that way back in the day, before Darwin came along, everyone believed that there was a divine creator, and everything was pretty copacetic. And then Darwin comes along and says, well, I found these different creatures. Specifically, the main one that people talk about is the birds that he found, called finches. And they all have different little varieties of finches. They're all about the same, but they have different beak shapes. And because they have a different beak shape, they eat a different kind of food. Some eat seeds, some eat little insects. And he believed that over time they were all one bird, and that over time they all sort of evolved into different kind of animal. Or, they're the same animal, but have a different kind of niche that they're filling with their different attributes. And that made pretty good sense to him and everybody else around him. And so that's kind of how the theory of evolution came about. And he said, well, if a little bird can evolve a slightly different beak shape that slowly evolves a slightly different diet, there's really nothing that says that you can't run that to some absolute extreme and that little bird also turned into a bald eagle or a lizard turned into a bird. I mean, that was his logic there. And the scientific world, it kind of made perfect sense, and to this day they more or less believe that. However, back in the late 1900s, a group of scientists got together and they started questioning that. And a lot of these scientists, they don't necessarily even had a Christian background. They just said, well, our science that we have on how any of this stuff comes to be isn't actually all that solid. Because really you can take Darwin's theory all the way back, but once you get to a single cell, the original cell, it doesn't really work. Darwin didn't have a whole lot to say on that, and there's really not a whole lot of information on how that single cell could come about. Because you really got to come from, once you get to there, the only thing smaller than that is you're coming from, volcanic ash. And you got to, if you're going to say that life on Earth originated from nothing, you got to explain how volcanic ash and stew turned into a single cell. And that's the jump right there. That's the real conundrum. And furthermore, one of the issues that that Darwin couldn't have explained because the science just wasn't advanced enough yet to know, was really what was going on inside of a single cell. Because back then they thought they barely even knew cells existed. By the 1950s, they were still even kind of thinking a single cell really there wasn't much going on inside of it. It was just sort of a little blob of goo, and that was about it. But after that, they started developing things like electron microscopes that could see cellular structures and things. And they realized the inside of a cell is way more complex than anyone had any recollection, right? It just blew everyone's minds because you have a whole world living inside of every single cell in your body, full of intricate, complex structures. And once you hit that level, then you run to this. There's a quote of Darwin where he says, if you could ever find any structure that is so complex in biology, it's so complex that you could not reduce it any farther to a sub-component and it still function, and you can prove that there's no way it could have just evolved that way, then the whole theory comes apart. And that was Darwin said that. And so once you get down to the cellular level, you end up coming across loads of these very small yet very complex structures that there's not really a way for them to evolve. You remove a single piece of it, the whole thing falls apart, and so evolution doesn't really function anymore. And so this is kind of what started all these scientists really questioning this. And they like I said, they all come together. They had a big conference because, a lot of these people, you start questioning the theory of evolution in a secular biology field, you lose your PhD real quick, right? You lose your professorship. So they all got together, had a quiet little conference out on the beach in California and said, are we all thinking the same thing here? And everyone was like, yeah, we are. And so they built this, those men eventually came together and they made that documentary that we showed to the senior youth last night. Trying to put together something for this morning, I actually managed to come across that same organization. They have a YouTube channel now. It's great. They have a lot more videos than just that documentary. And I actually got one of their videos. I'll play it today. We'll go ahead and play video 1. We got volume?
In Darwin's Fact Box in 1996, Behe spotlighted and made famous a number of really interesting discoveries that had been occurring in biochemistry and cell biology over the last 2 or 3 decades. And what biologists, molecular biologists, cell biologists, microbiologists have been discovering is that at the level of individual cells, there are little tiny examples of nanotechnology, little tiny machines at work. The flagellar motor is the one that Behe made most famous. It's a rotary engine that powers a whip-like tail, a protein tail, that functions like a propeller. And it moves the bacterium through liquid, enabling the bacterium to essentially track down its food supply. And this little machine includes a rotor, a stator, a drive shaft, a U-joint, bushings, bearings, and a whip-like tail that functions like a propeller. And the machine in some bacterial systems turns at 100,000 RPMs in one direction and can reverse direction on a quarter of a turn and turn 100,000 RPM in the other direction.
Bacterial flagellum is a true nanomachine, about 40 nanometers in size. It's amazing. I mean, E. coli, Salmonella, which are kind of our model systems for the bacterial flagellum, can propel a cell about 20 lengths per second through a very viscous medium, like water, to these organisms. And you extrapolate that to human scale. 20 body lengths per second, 6-foot person, times 20, 120 feet per second. Mark Spitz or Phelps would be setting records with this type of propulsion.
It's hardwired into a signal transduction circuit that allows the bacterium to sense changes in the sugar gradient in the surrounding liquid.
This signal transduction system is actually a short-term memory system where the cell is, if it's going in the direction of an attractant, a nutrient that it can use to metabolize, it follows that chemical gradient. If it's a repellent, it will sense that and move in the opposite direction. So it's more than just this engine.
It's an extraordinary piece of nanotechnology. It's high-tech in low life. And so just by spotlighting these extraordinary pieces of nanotechnology inside cells, and the flagellar motor wasn't the only one by any means, Behe, in a sense, opened up a window for people. He opened up the black box of the inner workings of the cell and said, look, this is much more complex than anything that the early evolutionary biologists had envisioned. Darwin knew nothing of this type of nanotechnology in cells, and at the very least, we've got to come up with an explanation for this.
Okay, so that's one example. That was kind of one of the examples that they used in the documentary last night. And you can see, they used the term drive shaft, stator, rotor. They didn't come up with that because they're trying to imply some sort of intelligent design. The scientists who discovered these structures, they look at that and said, no, that's what they are. They're literally those components. If you saw it in a car, that's what we would call it too. And they're incredibly complex. And any single one of those components, there's about 40 components that go to that structure. You remove any single one of them, the whole thing comes apart. There's no way you can really build that structure or that motor piecemeal. It doesn't evolve. It doesn't come about bit by bit. You would have to have the entire thing pre-built, loaded into the intelligence of that cell off the bat, or the thing just doesn't get built. There's no way that gets produced gradually, okay? And I mentioned, for the senior youth who are here, who are interested, I mentioned another thing called ATP synthase. We'll play that second video and you'll get to see that, because that wasn't in the documentary. You want to play video 2 for me?
All life on Earth depends on this tiny, energetic molecule, adenosine triphosphate, or ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA. Generating ATP for your living cells are mitochondria, electrochemical batteries that converts energy from the food you eat and oxygen from the air you breathe into ATP. So where does your ATP come from? Deep inside your mitochondria, rows of molecular motors generate ATP, the molecule essential to all life on Earth. Enzymes bring together reactants to form a chemical bond, converting mechanical energy into chemical energy. A ring of enzymes work in step, creating 3 molecules of ATP with each cycle. Inside the molecular motor, a rotating axle powers the sequence. The axle is attached to a rotary molecular motor, moved by the force of protons pushing from the other side of the membrane. A difference in proton concentration propels the molecular mechanism. Synthesis of ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA.
Okay, so these two structures, again, every single component of them they're completely essential for the function of life. Without that structure that we just saw there, life just doesn't exist. The cell dies. There's nothing even exists on a bacterial level if you don't have that. And you remove a single component from it, you change one component of it, everything's gone. There's no life without that. And it's incredibly complex. It kind of looks a little funny to our eyes because it's actually a 3D rendering of it, with a computer, but it's very accurate. I know the guy who made that, or I don't know him personally, but I know of him, the guy who made that animation. And he's a biochemist, and he uses really sophisticated machines to really figure out the structures of these molecules that do this. And he makes very accurate renderings of them so that we can see how they function mechanically. And that process that's going on there, it's far too complex of a thing to just happen on accident. Amen. Right? It's just not. You're not gonna. Each one of those, it kind of looks kind of like it's a bunch of like little globs stuck together. Well, each one of those globs is a function of amino acids sticking together, and it makes that structure. Well, it's incredibly complex, and those amino acids are originally organized in a big chain, and then those chains sort of congeal down into that blob, and then those blobs assemble into that big structure that you see. Well, the exact pattern in which those amino acids at the very ground level are assembled is incredibly important. If one of those is off, then the whole machine doesn't work. It has to be 100% accurate. And the pattern of that, of how those amino acids are put together, might be very similar to the way that we might maybe assemble an English sentence with letters. In the documentary, they use a great example. Imagine if you had, 2 lines from a famous Shakespeare play, right? To be or not to be. That is the question, right? It's just not very many letters. But what do you think are the odds if you just started dumping Scrabble pieces on a table, they're randomly going to assort into that sentence, right? It's not very likely, right? Technically maybe, but you're going to be dumping a lot of Scrabble pieces for a really long time. And even if you did, the complexity of what we just saw isn't just 2 sentences, it's several dozen volumes of Shakespeare plays stacked end to end. You're going to be really, dumping a lot of Scrabble pieces then, right? It's just not going to happen. No amount of time and random chance is going to make that happen. And so you wonder, okay, how does this happen? How does the cell know how to assemble something like that? Well, this is where we get into genetics. I'm going to use the one thing that I know if you're associated with the message at all, we kind of have a bad taste in our mouth around the word dogma, because Brother Branham uses it a lot, and not a very we'll say it, like complementary way. However, I do want to just give a definition of dogma because I don't want you to get hung up on that word because there's something really beautiful here if you can see it, and if my phone will unlock. Hang on. Oh, come on. All right, so the Oxford, or I'll go with the Webster's definition of dogma first. And it just simply means a doctrine or body of doctrines concerning faith or morals formally stated and and authoritatively proclaimed by a church. The Oxford definition of dogma would just simply mean a principle or set of principles laid down by an authority as incontrovertibly true. Okay, so this is just the absolute fundamental truth that any organization believes in. We might say that we would have a dogma, but our dogma would be the Bible, right? We're not going to accept anything outside of that. But in biology, they also have a dogma, and it is called the central dogma of biology. And the central dogma of biology states, describes the flow of genetic information in cells from DNA to messenger RNA to protein. It states that genes specify the sequence of mRNA molecules, which in turn specify the sequence of proteins. Now, that sounds like a lot of word salad. Hopefully this last video, I'll go with video 5. I'll skip over some of my videos. 5. The central dogma of biology. This is it. This is this dogma in practice.
The DNA double helix contains 2 linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
All right, so I got 4 minutes left, so we're going to try to wrap it up quick. So all of this really boils down to one thing, and that is inside of every cell, and I could condense the entire, we say what we call the central dogma of biology into one thing, and that is inside of every single one of your cells is a coded message. It is absolute. It includes the instructions to build your entire body through that process we just saw, building proteins. And pretty much your entire body is made out of these amalgamations of proteins, okay? And that genetic material, that, what is so often referred to in the message as that seed gene, right? We talk about genetics, a gene. We have genes within your DNA that code for proteins, that are the instructions on how to build proteins. That is intelligent. You can't really, that doesn't arise naturally. There's no way there's no known biological mechanism on how DNA could arise naturally. It is the instructions on how you're not just dumping Scrabble pieces on a table. It is the instructions on how to assemble those 23 different amino acids into something as complex as those structures we were seeing before, right? It's the blueprints. It is the absolute proof that some intelligence built that, because it's so specific. It can't really happen through random chance. And if we saw this in any other format, right? Bill Gates called DNA the most complex computing system known to man. And this is Bill Gates, right? He writes computer programs, and he doesn't, rely on random chance to do it, right? Computer programs are built through intellect, not through random chance. And so if you have anything that is very clear written instructions on how to produce something that complex, you're not going to call that random chance. You're just not. It doesn't make any sense, any more than we would see, a statue carved out of rock and say, well, it's clearly wind erosion, right? It's just not gonna, that doesn't make any sense. Obviously, it's a person carved out of stone, so some person must have carved it. Well, this is a person written in an intelligible code. We might not understand all of it, but it's very clearly build structures that turn into a person. So we're not going to call that random chance either. It's very clearly built by an intellect. It's very clearly made by an intelligence. And so that was about the extent of the documentary. And where I would like to go from there is, I'll just say this. That the genetic material that we saw, right, that process of DNA, and they mentioned that it's made up of 4 letters, A, C, T, and G, right? It's just repeating lots of different patterns of those 4 letters repeated across these long strands of DNA. Those in a human body, there are, 3.2 billion in a single set of human DNA, 3.2 billion of those little 4 letters in a line, right? And that's what makes up the whole body. And your body can uncode those processes or those sequences and turn it into a person, much like a computer can turn ones and zeros and turn it into a picture, right? Very similar. It's not too far off from the same idea. With 3.2 billion of those letters strung together, you can't have one of them be wrong. Can you pull up that picture, the Brother Mark, pull up the picture right at the end that I had there? This is sickle cell anemia. So you see on the left, normal red blood cell. That's what everyone's got flowing through your veins and your arteries. That's what they're supposed to look like. On the right, you have a sickle cell. People with sickle cell anemia, their red blood cells look like that. And you can tell that's not a good thing, right? The shape of your red blood cell is what helps it move through your arteries without getting caught on things and getting stuck. It helps with the absorption of oxygen and for it to do its job. Well, if it's shaped all like a crescent moon, it gets hung up in your arteries, creates blood clots. It's not going to absorb oxygen correctly. It's going to cause a lot of problems. Now, that major twist in the entire structure of a red blood cell comes from a single letter being off. Just one. That's all it takes out of 3.2 billion, right? And this is, when Brother Branham talks about, the spoken word being the original seed, and what's at the bottom of a seed is just a genetic package. So what's at the heart of a seed is written instructions on how to manifest the life that seed is designed to manifest. It's the DNA inside of it. You get one letter off, you get a disaster, right? You get, we have the Word of God. This is our this is our genetic package that tells us how to manifest into God's life, right? that spoken word was the original seed that will manifest into God's life. Well, you get one letter off in that, you will end up with something that is not God's life. It'll be a disaster, right? So, I'll wrap it up there. We're about 1 minute over, so, and Brother Matt already told me I could have a second one, so you'll get a sequel. All right, we'll stand and have a word of prayer. Father, we come before you this morning and want to thank you for this opportunity to maybe say something that would help the people, maybe that they would see you in nature more, and even see you within themselves, Father. That maybe they would take these things and it would help them with their walk with you, Father. And Father, please, I would ask that you would take these words that I would say, that any nerves or any spirit of myself that wouldn't extort or contort what you would have to tell the people, and they would hear it clearly through what you would have to say, Father. We ask these things in Jesus' name. Amen. And we'll have church in about 10 minutes.